After studying this module, you shall be able to know what is central dogma, how gene is expressed, what are the various steps followed in the process of transcription and translation, what is genetic code. Gene expression. Gene expression is the process by which information from a gene is used in the synthesis of a functional gene product. These products are often proteins, but in non-protein coding genes such as transferred RNA or small nuclear RNA genes, the product is a functional RNA. The process of gene expression is used by all known life forms, eukaryotes, including multicellular organisms, prokaryotes like bacteria and archaea, and viruses, to generate the macromolecular machinery for life. Several steps in the gene expression process may be modulated, including the transcription, RNA splicing, translation, and post-translational modification of a protein. Gene regulation gives the cell control over structure and function and is the basis for cellular differentiation, morphogenesis, and the versatility and adaptability of any organism. Gene regulation may also serve as a substrate for evolutionary change, since control of the timing, location, and amount of gene expression can have a profound effect on the functions or actions of the gene in a cell or in a multicellular organism. In genetics, gene expression is the most fundamental level at which the genotype gives rise to the phenotype. The genetic code stored in DNA is interpreted by gene expression, and the properties of the expression give rise to the organism's phenotype. Such phenotypes are often expressed by the synthesis of proteins that control the organism's shape or that act as enzymes catalyzing specific metabolic pathways characterizing the organism.
One gene, one polypeptide hypothesis. The hypothesis that a large class of structural genes exist in which each gene encodes a single polypeptide, which may function either independently or as a subunit of a more complex protein. Originally, it was thought that each gene encoded the whole of a single enzyme, but it has since been found that some enzymes and other proteins derive from more than one polypeptide and hence from more than one gene. Central dogma. Central dogma is the process in which DNA is converted into RNA and RNA further synthesizes protein through transcription and translation process respectively. In 1958, Francis Crick recommended the central dogma, but there is one exception. In retrovirus, single-stranded RNA is converted into double-stranded DNA with the help of enzyme reverse transcriptase. RNA and its types. Ribonucleic acid, or RNA, is a polymeric molecule. It is implicated in various biological roles in coding, decoding, regulation, and expression of genes. DNA and RNA are nucleic acids and, along with proteins and carbohydrates, constitute the three major macromolecules essential for all known forms of life. Like DNA, RNA is assembled as a chain of nucleotides, but unlike DNA, it is more often found in nature as a single strand folded onto itself rather than a paired double strand. Cellular organisms use messenger RNA or mRNA to convey genetic information using the letters G, A, U, and C to denote the nitrogenous bases guanine, adenine, uracil, and cytosine that directs synthesis of specific proteins. Many viruses encode their genetic information using an RNA genome. Some RNA molecules play an active role within cells by catalyzing biological reactions, controlling gene expression, or sensing and communicating responses to cellular signals. One of these active processes is protein synthesis, a universal function whereby mRNA molecules direct the assembly of proteins on ribosomes. This process uses transfer RNA or tRNA molecules to deliver amino acids to the ribosome, where ribosomal RNA links amino acids together to form proteins. There are three types of RNA: mRNA, messenger RNA, or mRNA, carries information about a protein sequence to the ribosomes, the protein synthesis factories in the cell. It is coded so that every three nucleotides, a codon, correspond to one amino acid. In eukaryotic cells, one precursor mRNA or pre-mRNA has been transcribed from DNA. It is processed to mature mRNA. This removes its introns, non-coding sections of the pre-mRNA. The mRNA is then exported from the nucleus to the cytoplasm, where it is bound to ribosomes and translated into its corresponding protein form with the help of tRNA. In prokaryotic cells, which do not have nucleus and cytoplasm compartments, mRNA can bind to ribosomes while it is being transcribed from DNA. After a certain amount of time, the message degrades into its component nucleotides with the assistance of ribonucleases. tRNA. Transfer RNA or tRNA is a small RNA chain of about 80 nucleotides that transfers a specific amino acid to a growing polypeptide chain at the ribosomal site of protein synthesis during translation. It has sites for amino acid attachment and an anticodon region for codon recognition that binds to a specific sequence on the messenger RNA chain through hydrogen bonding. rRNA, that is, ribosomal RNA, is the catalytic component of the ribosomes. In the cytoplasm, ribosomal RNA and protein combine to form a nucleoprotein called a ribosome. The ribosome binds mRNA and carries out protein synthesis. Several ribosomes may be attached to a single mRNA at any time. Nearly all the RNA found in a typical eukaryotic cell is rRNA. Transcription. Transcription is the first step of gene expression in which a particular segment of DNA is copied into RNA, that is mRNA, tRNA, or rRNA, by the enzyme RNA polymerase. Both RNA and DNA are nucleic acids, which use base pairs of nucleotides as a complementary language. The two can be converted back and forth from DNA to RNA by the action of the correct enzymes. During transcription, a DNA sequence is read by an RNA polymerase, which produces a complementary anti-parallel RNA strand called a primary transcript, that is, DNA is read from 3' to 5' end during transcription, whereas the RNA is formed from 5' to 3' end direction. Ingredients for transcription. The following components play a significant role in transcription. First is RNA polymerase enzyme. It was discovered by Charles Louis, Audrey Stevens, and Gerard Hertwitz in 1960. It is an enzyme useful for creating RNA chains using DNA as a template. And second is DNA strand. Steps involved in transcription. Mainly three important steps are involved in the process of transcription. First is initiation. RNA polymerase bind to the promoter site of DNA regions. The promoter consists of initiation site where transcription occurs. RNA strand is released then. Next is elongation. During this process, RNA polymerase adds nucleotides to the 3' end of the growing RNA. It also involves proofreading mechanism. And lastly is termination. Once RNA polymerase enzyme reaches termination site of DNA, then no more nucleotides are added. RNA chain is released, and at this point, transcription ends. Translation. Translation is the final step on the way from DNA to protein. It is the synthesis of proteins directed by a mRNA template. The information contained in the nucleotide sequence of the mRNA is read as three-letter words or triplets called codons. Each word stands for one amino acid. During translation, amino acids are linked together to form a polypeptide chain, which will later be folded into a protein. The translation is dependent on many components, of which two are extra important. First of all, the ribosome, which is the cellular factory responsible for the protein synthesis. It consists of two different subunits, one small and one large, and is built up from rRNA and proteins. Inside the ribosome, the amino acids are linked together into a chain through multiple biochemical reactions. The second component is the tRNA, a specialized RNA molecule that carries an amino acid at one end and has a triplet of nucleotides, an anticodon at the other end. The anticodon, of a tRNA molecule can base pair, that is, form chemical bonds with the mRNA's three-letter codon. Thus, the tRNA acts as the translator between mRNA and protein by bringing the specific amino acid coded for by the mRNA codon. Ingredients involved in translation. The ingredients required for the synthesis of translation are ribosomes, tRNA, mRNA, aminoacyl tRNA synthetase, and genetic code. Ribosomes in detail. The ribosome is a large and complex molecular machine found within all living cells that serves as the site of biological protein synthesis or translation. Ribosomes link amino acids together in the order specified by messenger RNA or mRNA molecules. Ribosomes consist of two major components: the small ribosomal subunit, which reads the RNA, and the large subunit, which joins amino acids to form a polypeptide chain. Each subunit is composed of one or more ribosomal RNA molecules and a variety of proteins. The ribosomes and associated molecules are also known as the translational apparatus. The sequence of DNA encoding for a protein may be copied many times into RNA chains of a similar sequence. Ribosomes can bind to an RNA chain and use it as a template for determining the correct sequence of amino acids in a particular protein. Amino acids are selected, collected, and carried to the ribosome by transfer RNA or tRNA molecules, which enter one part of the ribosome and bind to the messenger RNA chain. The attached amino acids are then linked together by another part of the ribosome. Once the protein is produced, it can then fold to produce a specific functional three-dimensional structure. A ribosome is made from complexes of RNAs and proteins and is therefore a ribonucleoprotein. Each ribosome is divided into two subunits: a smaller subunit which binds to the mRNA pattern and a larger subunit which binds to the tRNA and the amino acids. When a ribosome finishes reading an mRNA molecule, these two subunits split apart. tRNA is attached to A site of ribosome, whereas new polypeptide is released from the B site of ribosome. mRNA is attached to ribosome through binding site. tRNA, a transfer RNA, is an adapter molecule composed of RNA, typically 76 to 90 nucleotides in length, that serves as the physical link between the nucleotide sequence of nucleic acids and the amino acid sequence of proteins. It does this by carrying an amino acid to the protein synthetic machinery of a cell or ribosome as directed by a three nucleotide sequence codon in a messenger RNA or mRNA. As such, tRNAs are a necessary component of protein translation, the biological synthesis of new proteins according to the genetic code. The specific nucleotide sequence of an mRNA specifies which amino acids are incorporated into the protein product of the gene from which the mRNA is transcribed, and the role of tRNA is to specify which sequence from the genetic code corresponds to which amino acid. One end of the tRNA matches the genetic code in a 3-nucleotide sequence called the anticodon. The anticodon forms 3 base pairs with the codon in mRNA during protein biosynthesis. The mRNA encodes a protein as a series of contiguous codons, each of which is recognized by a particular tRNA. On the other end of the tRNA is a covalent attachment to the amino acid that corresponds to the anticodon sequence. Each type of tRNA molecule can be attached to only one type of amino acid. So each organism has many types of tRNA. In fact, because the genetic code contains multiple codons that specify the same amino acid, there are several tRNA molecules bearing different anticodons which also carry the same amino acid. mRNA. mRNA sticks itself to the binding site of ribosomes, then matches the codon with tRNA in order to synthesize a specific protein. The mRNA coding for a particular codon is coupled with tRNA, which complementarily codes for a particular amino acid. The mRNA codes for a particular codon AUG, complementary to which tRNA produces UAC anticodon, resulting in the synthesis of a specific amino acid called methionine. Aminoacyl tRNA synthetase. Aminoacyl tRNA synthetase is an enzyme that catalyzes the esterification of amino acid. This enzyme is used during translation process for the synthesis of proteins. tRNA attaches to specific amino acid by covalent bond by aminoacyl tRNA synthetase enzyme. Genetic code. A genetic code is a triplet sequence of bases found in mRNA. Each code specifies each amino acid. There are 64 different combinations of bases which code for amino acid. Among those combinations, there are 61 sense codon and 3 non-sense codon, that is UAA, UAG, and UGA. If one base represented each amino acid, it will produce 4 different combinations of A, T, G, and C. If 2 bases represented each amino acid, it will produce 16 different combinations. If triplet bases represented each amino acid, it will produce 64 different combinations. Steps involved in translation. There are 3 steps involved in translation process, which is initiation, elongation, and termination. First step in the process of translation is initiation. Firstly, the mRNA attaches itself to a small subunit of ribosomes at the initiation codon sites, and first tRNA attaches. Secondly, large subunits of ribosomes attaches with the tRNA at P site. The mRNA, which carries the codon, is attached to a small subunit of ribosomes at initiator site. tRNA is also attached to a small subunit of ribosomes which carries anticodon. Next, large subunit of ribosomes attaches to tRNA through P site. Second step is in translation is elongation. During elongation, tRNA attaches by its anticodon to the codon of mRNA to its A site. The mRNA is attached to a small subunit of ribosome. mRNA carries the codon AUG respectively. tRNA couples with mRNA carrying anticodon UAC for that codon which is complementary. Secondly, as soon as the correct coupling of mRNA and tRNA takes place first, peptide bond is formed as new amino acid arrives. The first amino acid to be released is methionine. Next, as the first amino acid is formed, next coupling of tRNA and mRNA takes place to produce another amino acid and a peptide bond is formed. tRNA leaves the P site of ribosome and the ribosome moves down the mRNA. The last stage of translation is termination. As soon as elongation of amino acid reaches the stop codon or nonsense codon, that is UAA, UGA, and UAG, the termination process occurs and then synthesized protein is released. The process of elongation stops when the ribosome reaches the stop codon. The components of ribosome disassemble due to the release factor and a protein which is synthesized is released.
Gene expression. It is a process in which the information carried by the genes are used in the formation of its products. One gene is responsible for the synthesis of one polypeptide. Central dogma is the process in which DNA gets transformed into RNA, while RNA further synthesizes proteins by undergoing transcription and translation processes respectively. There are three different types of RNAs known: mRNA or messenger RNA, which encodes amino acid sequence by attaching itself to ribosomes to synthesize proteins; tRNA or transfer RNA, which transports specific amino acids to ribosomes for protein synthesis; rRNA or ribosomal RNA, which gets associated with protein to form ribosomes. A specific segment of DNA is transcribed or copied to RNA, which further encodes at least one gene, is transcription. Three main steps involved in the transcription process. Initiation. RNA polymerase binds to the promoter site of DNA regions. The promoter consists of initiation site, where transcription occurs and RNA strand is released. Elongation. During elongation, RNA polymerase adds nucleotides to the 3' end of the growing RNA. It also involves proofreading mechanism. Termination. Once the enzyme RNA polymerase reaches termination site of DNA, then no more nucleotides are added. RNA chain is released and transcription ends. The process in which protein synthesis takes place with the help or under the direction of mRNA is translation. Genetic code. It is the triplet sequence of bases found in mRNA. Each code specifies one amino acid. There are 64 different combinations of bases which code for amino acid. Among those combinations, there are 61 sense codons and 3 non-sense codons, which are UAA, UAG and UGA. There are 3 steps involved in translation process, namely initiation, elongation and termination.